looking at Unit 7, which is Chapter 8 in your OWL book, and Chapters 11 and 12 in the Penguin book if you happen to have that copy. Chapter 8 is about cell reproduction or making new cells, and the most important thing to think about when cells are dividing is managing your DNA. Remember, your DNA is a molecule of life. It has all of the genetic information, every single set of instruction for making every single part of your body is in your DNA. And if I'm going to divide my cell, I need to make sure that I'm taking care of handling this very fragile molecule as I move it into two new places. So normally, when your cell's just being a cell and it's just chilling, it's in this long stringy form that's called chromatin, and that way it can be easily read, it can be used by the cell, so it can make all those parts that are needed without causing too much problem. But moving a tangled mass of DNA isn't going to work well for our process of cell division. So what your cell will do is it's going to pack it up. It's going to take that DNA and it's going to start coiling it up, forming these things called chromosomes. And you can see over here in the picture they look like little X's, and you can see how much easier that's going to be to just like, oh, I can just pick up an X and move it over here, or I can put something, I can move it and put it over here. It makes it a lot easier. So it forms these chromosomes so that when we want to move our DNA into new homes, it's much easier for transportation. So how does your cell wrap up the DNA? Well, it's going to use these proteins called histones. And I always imagine the histones like hair curlers, and that I'm just wrapping my hair around a curler to help hold it in place. So basically what we're doing is we're just wrapping our DNA around these histones to hold them in place. And that's how I can take this huge long molecule, which is like 5.5 feet long, and shrink it up into a teeny tiny little package for cell division. So make sure you learn these terms. You're going to be hearing them a lot, and it gets easy to confuse. It's almost like we're saying the same thing in 5 different languages, going DNA, genes, chromatin, chromosomes, chromatid. It can get pretty confusing. So this is where knowing your vocabulary is going to really help you out in the long run. So here is a chromosome, and like I said, because chromosomes hold your DNA, then they have a really important function. Their job is to keep that information safe, and another neat thing is it actually lets us identify where certain genes are located on the chromosome itself, because as you see as I wind this up, it makes this really cool pattern, and those lines on it are called bands, and they're actually specific to different genes in your body. And so that's how we start locating different genes in your body, is by where they're at with that banding pattern. So when we look at the overall structure of chromosomes are going to be formed after the DNA is copied. Because if I'm making 2 new cells, well then each new cell needs a copy of the DNA, or it's not going to work right. So basically, it's like I took my DNA to a printer and I printed it off and I laid my 2 copies together. And those 2 copies are called chromatids. They're 1/2, if you can see it here, 1/2 of each chromosome is a chromatid. Not top and not bottom, but the sides, left and right, are your chromatids. And because they're copies, they're identical to each other. I don't want different information in different cells. I want the cells to have the same information, which is why I copied it. And I think the cell organizes itself with this copy, much like we staple paper together. They staple the chromosomes together right here in the middle with this protein disc called a centromere. So that way I have my copies and I'm holding them together, and now I'm really neat and organized in keeping my genetic information in this little bundle all tied in together, so I'm not going to shift stuff around and mess up transferring it into their new homes, the new cells that we're hopefully going to make. Now chromosomes are different in different types of cells. Prokaryotic cells actually have like maybe a single chromosome, and it's circular, where when we look at ours, it's a big long stringy helix thing. It coils up into an X. Theirs is just a single ring that sits right up next to the plasma membrane. It also has like little extra plasmids, which are made out of DNA, so it's like bonus DNA, and those extra plasmids help the bacteria develop antibiotic resistance. It's like it gives them like I don't know, protection or a power level or something crazy like that. So if you look at your diagram of a prokaryotic cell, this is the DNA all snuggled up against the plasma membrane, and here are the little plasmids there that kind of act as wingmen to help give it some bonus information so that it can fight against different antibiotics that we might use to kill that bacteria. In our cells, in eukaryotic cells, the diversity is tremendous, and the thing to keep in mind is not how many chromosomes you have that matters, it's how big your chromosomes are that matter, because the bigger your chromosome, the more information it's going to hold. For example, we only have 46 chromosomes, that's it, but a fern, which is a plant in our example, has over 1,200. But they're short and they're fragmented and they don't hold as much information as ours do, and so we're more complex than a plant. The little guy over here in the picture is a fruit fly. I think, off the top of my head, they only have like 11 chromosomes, so they're fun for genetic research and study, and we'll actually kind of mention them off and on again. There are two types of chromosomes. There are sex chromosomes. Those chromosomes will determine whether or not you will be born male or female. And in biology, when we talk about male, female, it's not gender. Gender is a social construct. We're just looking, were you born female, were you born male? And that's just how we look at it in terms of a biological perspective. But these are the chromosomes that are going to decide if you are male or female, the X and the Y. Now remember, before we said there was 46, so this is just 2 of the 46. The other 44 chromosomes are what we call your body chromosomes, and they are technically called autosomes. So all the other information that's going to make up you, your hair color, your eye color, the shape of your teeth, the color of the nose hairs, those are the things that are going to be found on these chromosomes. So where do you get your chromosomes? Well, you get them from your mom and you get them from your dad. Half the information comes from mom, half the information comes from your dad. So when you look at that, your chromosomes are going to match up then. Dad's chromosomes, mom's chromosomes are going to match up, and that's what we call homologous chromosomes. Because I need the information that, say, codes for your height to match up with the chromosome that also from your dad codes for your height. So they're going to be the same size and shape. They're going to have that same pretty banding pattern. And the neat thing is after conception, when the sperm meets the egg, there's a 24-hour window where your chromosomes find their homologous pair, and then they match up. And then once that is completed, it's like go from there. You get rapid cell division, how we start forming all the cells of your body, and it's a pretty neat thing how they can just like find their mate basically and start going to work. Because if I'm going to make any part of your body, I need to have both copies of the gene in order to do it. I can't do it with just one copy. You gotta have a full set, and that's why you get half of that set from mom, half the set from your dad. When we want to look at your chromosomes, what they'll do is they'll find a cell that's kind of undergoing cell division, and that's when the DNA is going to be all tucked up and condensed, and they'll take a picture of it, and then they'll cut them out and they'll match them up. Of course, they have computers for that now, and that lets us look at what chromosomes they have, how many they have, are there any problems with their chromosomes. So I can look at chromosomal abnormalities, but it won't let us look at genetic level mutations because that's something wrong with the DNA itself. But a karyotype will let us have like a big picture of it. So here's an example of a karyotype. You can see that the pretty banding color, you can see that they're the same size, same shape, and at the very end of a karyotype we put their sex chromosomes. So 1 through 22 are your autosomes, 22 pairs, total 44. Last two, 45, 46, will give us the sex of the individual. And 2 X's means you're a girl, so we have an XX, and an XY would mean you're a boy. Moms can only give their children an X, so it's the dads that determine if you're going to be born male or female. So I'm going to pause here in our video so that we can work our way into who killed Captain Relish.